A point mutation within a DNA sequence occurs whenever one nitrogenous base is substituted for another. There are two different types of point mutations, and these are transitions and transversions. Transitions happen when a purine is substituted by another purine, or a pyrimidine is substituted by another pyrimidine. Here, I have the purines adenine and guanine written in green, and the pyrimidines thymine and cytosine written in pink. I have labeled all of the possible substitutions for a transition mutation. Transversions are different from transitions in that a purine is replaced with a pyrimidine or vice versa. I have also written the possible substitutions for this type of mutation. Point mutations can have several different effects on the amino acid sequence, which in turn can alter the function of the proteins. There are 4 different specific types that I'll be discussing, and these are missense, nonsense, silent, and read-through mutations. Here we have an example of a missense mutation. This type of mutation takes place when the nitrogenous base is replaced with another nitrogenous base that allows for the new codon to code for a completely different amino acid. We can see that whenever we substitute the A highlighted for a G, the new codon becomes AGC, which codes for serine. This is also an example of a transition mutation because the purine adenine is substituted by the purine guanine. Next, we have a nonsense mutation, and this mutation occurs whenever the change in a single nucleotide causes a specific codon to become a stop codon. In this example, the C in the codon becomes an A in the mutant. The new codon is TGA, which translates to the stop codon UGA. This is also an example of a transversion because the pyrimidine cytosine was substituted with the purine adenine. Silent mutations happen whenever there is a change in a single nitrogenous base, but the new codon still codes for the same amino acid. In the mutant sequence shown, the C is substituted with a T. The original codon, TGC, codes for the amino acid cysteine, and the codon TGT also codes for cysteine. Lastly, we have a read-through mutation. This type of mutation occurs whenever the nucleotide change occurs within the stop codon and the new codon codes for an amino acid. This results in a longer protein chain. The codon TGA that originally coded for the stop codon is now TGG, which codes for the amino acid tryptophan. Now, translation continues, and CCC translates to proline, and GGC translates to the amino acid glycine. This is also an example of a transition mutation because the purine adenine is substituted by the purine guanine.